Hello, Harish sir.
Pramod, good morning.
Good morning, sir. Sir, we start the session.
Hello.
Start the session in another 5 minutes.
Okay. Whether my audio is clear?
Yeah, it's very clear, sir. It's very clear. Everything is fine.
Okay, then fine.
Thank you, sir. Hello, Shankar sir. Is it I'm audible?
Hello.
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 Hello. Yeah. The voice is clear. Thank you.
Hello.
Hello.
I'm not hearing your voice. Is my voice is clear?
Yeah, it's clear. It's clear.
Okay, fine. Fine.
Very good morning to all of you. I welcome you all for the 6th day session of the National Webinar Series 2020 organized by Department of Biochemistry, Davangere University, and Society of Biological Chemists, Davangere chapter. And the webinar, the theme of the webinar is immunity and infection, biochemical approaches to therapies. Now, I request one of our colleague, Dr. Shankar, Associate Professor, Department of Technology, Davangere University, to introduce the speaker of the first session, Dr. Harish Prashanth, who is a senior scientist at CSIR-CFTRI, Mysuru, and he is going to speak on polysaccharides' role in immunology. Shankar sir, please.
Thank you, Dr. Pramod. Good morning, one and all. First of all, I would like to thank you, congratulate Department of Biochemistry, Davangere University, Davangere, to organize this National Webinar Series 2020 on immunity and infection, biochemical approaches to therapy. So, and also I'm very glad to introduce today's first speaker, Dr. KV Harish Prashanth, whom I know since from 2 decades as a senior colleague, a friend, and advisor. So Dr. KV Harish Prashanth, a senior scientist in the Department of Biochemistry, Central for Technological Research Institute, CSIR-CFTRI, Mysuru, India, has obtained his MSc in Biochemistry from University of Mysuru, and PhD in Biochemistry from CSIR-CFTRI under the supervision of Dr. Taranathan, a distinguished scientist in carbohydrate research. He did postdoctoral research in Indian Institute of Science, Bangalore, and later in the Division of Biotechnology, Linköping University, Sweden, in the project entitled Microarray for Genomic Profiling of Metabolic Load during Protein Expression, where he was one of the leading investigators for his project with Professor Nagaraj CF. He worked extensively on cytosome, especially with respect to structure-function relationship, biochemical and biotechnological aspects, as well as polymeric blending studies for active and smart food packaging application, and also shown cytosine derivatives as potent anti-angiogenic as well as anti-tumor, possible protection from neurodegeneration and lifestyle disorders by stabilization of against antioxidants. As evident from his publication in peer international journals with more than 2,800 citations. Apart from this, he has studied other carbohydrates like gums, resistant starch, fructans, pectin modifications, and fortification studies for food applications and transfer technologies to industries. Apart from various recognition and honors, in 2016 bestowed with Dr. M. Mahadevia National Award with Silver Plaque, instituted by All India Food Processors Association, New Delhi, and recently in September 2019, awarded Best Faculty Researcher Award 2019 with Cash Award constituted by Indian Chitin and Cytosine Society at Indian Chemical Technology, Mumbai. Involved in studying the following carbohydrate-related areas: studies related to neurotoxicity, neurodegeneration, tumor biology, and other lifestyle disorders like diabetes and relative intervention of dietary functional polysaccharides in respective amelioration. Effect of bioavailability of carotenoids induced by cytosine derivatives, characterization of nanoparticles for food applications, standardization of process condition of polysaccharides based coating formulations for shelf life extension for fruits, studies on elicitation of plants for secondary metabolite production using cytosine based derivatives, increasing bone strength by water soluble low molecular weight cytosine and cytosine oligosaccharides with specific degrees of acetylation and polymerization. Apart from being a researcher, Dr. Harish Prashanth sir. Presently, he is an advisor at Adi Chunchandagi University in Nagamangala, Karnataka. Presently, he is a Honorable Secretary of Association of Food Scientists and Technologists, that is AFSTI, headquarters India, and Honorable Secretary of Society of Biological Chemists of India, Mysuru chapter, and Central Executive Member for Association of Carbohydrate Chemist and Technologist, India. With this brief introduction, I will welcome Dr. Harish Prashanth sir to speak on his title of the talk today, Polysaccharides, Role in Immunology. Please welcome, sir. Over to you.
Thank you, Dr. Shankar, for that very exhaustive introduction of me. And I welcome you all, and good morning to one and all over there. And first, at the outset, I want to thank Vice Chancellor, the head of the Department of Biochemistry, Tumkur University, and all the faculty members who are directly and indirectly involved in this webinar series. So with this, I'll straight away go to today's topic. This is all about the immunoregulation of polysaccharides. So which here in the first slide which I have given is about, it constitutes of whole summary of my talk today. So we know that there are several biomolecules which can induce immunomodulatory activity. As such, polysaccharide is the one which has recently gained a lot of interest. So with a lighter introduction of immunology, we know that immune system is mainly because of it has to defend against the infection and the diseases. So this is the one the whole immune system why we require. And it consists of, you can see in the slide, it consists of two major arms. One is innate immune system and one is adaptive immune system. So it comprises of many cells with its own very special function, and as well as it also works together. So generally it fights of a defense. I mean, fights to defend the disease or the intervention of pathogens whenever it is required at that time. So all of the cells system is like it develops from stem cell, if we all know that, and located in which is located, the stem cells which is located in a bone marrow, and give rise to like lymphoid as well as myeloid progenitor cells, which in turn each of which differentiates with varieties of cell types. So one which leads to lymphoid progenitor differentiate into three major cells: beta cells, T cells, and N killer cells, that natural killer cells. And when it comes to myeloid lineage, it gives rise to innate immune system, where it it is also like which like myeloid which gives you innate, and well as lymphoid progenitor which leads to adaptive system. So when it comes to myeloid, it is a it's all innate immune system which is being given. I don't go into the detail. I'll go to straight away the next, which is a difference between innate immune system, where the macrophage will recognize these ligands. Ligands means the pathogen-associated molecular patterns. This pattern recognition receptor recognize this, and immediately intervene into pathogen phagocytosis, and it eliminates from the body. And this further leads to adaptive immunity after a little longer time by inducing the T cells and B cells thereby. So this is a type of immune response, if you see. And one more thing is about one which leads in the same way, which leads to cytotoxic T cells. It's a cell-mediated immunity. If it activates the B cells, and B cell in turn makes antigen-antibody complexes, which leads to humoral immunity. So I think these are all the things already been dealt by so many speakers previously. This is an overall view of immune response. Here, I'll take a little while to explain this as it is the one which gives our insight about our work later on. So macrophage, whenever it is in contact with the antigen or the ligand, it engulfs and digests the same. And the part of the antigen fragment will be presented on to the surface by these macrophage cells, which will be recognized by the T cell, and this T cell gets activated, and the activated T cell either it make a way towards a cell-mediated immune response as well as humoral immune response. So this we know that T cell when it proliferates into T helper 1 cell or T helper 2 cells, which also secretes a lot of cytokines. So here the crosstalk happens between between the cells of macrophage and T helper cells, and in return to produce more and more of this kind, as well as the proliferation occurs. So when it goes to T helper cell, T helper 1 cell, which leads to cytotoxic T cell in cell-mediated immune response, recognizes the infected cells, which through MHC1, and it's tried to eliminate by making this cell to destruct, and destruction of this cell leads to elimination. And again, in another humoral immune response, what happens, like Th2 cell, it interacts with the B cell through co-stimulators, and this in turn releases after maturation or proliferation and also differentiation, produces a lot of antibodies. So here, to make this much of antibodies, here the antibodies, we know that 5% of this is constitute of carbohydrates, and this is the key, like why carbohydrates are very important for the body. And whichever thing you say, even the antigen or the recognition receptors, everything is a sort of glycoprotein, and this part of carbohydrate moiety will be there all the times. So for this reason, I have just given an overview of immune responses, how it happens whenever there is a antigen being circulated in the blood. And next we'll move on to the carbohydrate importance. I'll just give you the glimpse of the carbohydrate importance especially for the students fraternity, where we know that first line of energy trapped in this world through sunlight is, and which will convert into a carbohydrate, that is glucose. How? By virtue of entrapping these energy sunlight through these green plants, which produces this carbohydrate. So that's how the carbohydrate journey starts. And you know, all the metabolism with respect to plants and animals, without carbohydrate, you cannot proceed any metabolic activities. So it's a central, it has gained a central importance in all the metabolism of plants and animals. Not only functionally or physiologically it is important, but also it is a structural component of plants and animals, even the bacteria, that is microorganisms. So everywhere the carbohydrate is present. The energy being stored always either in plants or animals in the form of carbohydrate. That is, we know we store our energy by virtue of glycogen, and plants will store the energy in the form of starch. So with all these things, even for it is very important for immune responses. As I told you, either it's an antigenic determinants or an antibody structure, the carbohydrate is the important component. Next, we'll move on to how the polysaccharide is important, like the maximum or the most abundant form of biomolecule is a polysaccharide. And this polysaccharide is naturally macromolecules which constitutes of monosaccharides. So monosaccharides units leads to polysaccharide. And this polysaccharide might be a homopolysaccharide or a heteropolysaccharide, depending on the monosaccharide. If it is the same monosaccharide, like if it is a glucose, it leads to amylose. If it is like having a different like hyaluronic acid, chondroitin sulfate, these are all the heteropolysaccharides. So there are a distinguish between hetero and homopolysaccharide. There is a wide range of this sort of polysaccharide. Unlike protein, the sugar differences is so huge that any change in small functional groups or structure leads to different kinds of polysaccharide compounds. So till now, it has been more than 300, which has been identified. Still, it's going on. It is, as I told you, it is present in all animals, plants, microorganisms, and engage in all sorts of variety of physiological functions. So when this polysaccharide become very popular with respect to immunology, means around 60s, like 1950s to 60s, where this main focus on immunomodulation of this polysaccharide took place. Continuing with some of the importance with respect to immunomodulation of this polysaccharide. So the plant polysaccharide especially got a much attention, because microbial polysaccharide has some issues in the sense it has some contamination of endotoxin. Along with this, always the issue was there. So there is the same way of immunomodulation with respect to polysaccharide is a plant polysaccharide. So today I'll be more focusing on this plant-based polysaccharide and its immunoregulation aspects. So how this polysaccharide will give such response? So this is like by it activates the macrophage and enhance the immune responses. So how again it is through increasing the ROS production, and also in turn it also has a lot of studies been done, and they have shown that it secretes a lot of cytokine and chemokines. So that's why even the plant polysaccharide now is being thought for therapeutic potential towards immunomodulatory medicine. So a variety of polysaccharides have been discovered, and their immunomodulation activity has been elucidated, the structure and function relationship somehow. And sorry for this slide, which many of you can't see it. Uh, this is just to represent, like, there is a lot of work, especially plant polysaccharide, they're doing it. And here, with respect to different molecular weights or different like monosaccharide composition, there is a huge difference between the function and its structure. And also with respect to the bond it is constitute of. If it is a alpha 1,6, beta 1,4, or beta 1,6, whatsoever, the bond also, even the smallest small anomeric change will change the functionality of this carbohydrate or a polysaccharide. So that is how the important has been gained for the biological activity of this plant polysaccharides. So next is I'll just touch in one slide about modification of this polysaccharide, why it is required to enhance the bioactivities. See, sometimes the polysaccharide, you know, it's of high molecular weight, and there will be solubility issues, and also there will be a huge viscosity should be encountered whenever you wanted to use as a bioactive molecules. So for that to reduce or to change, we do modification for this polysaccharide. And this modification was basically based on the structure function relationship. So further, the molecular modification, whatever we do, is by three methods, like chemical method, physical method, or biological modification. So I'll just show you how these three modification will be done. Like for example, if it is a physical modification, we do use ultrasonication or gamma irradiation. Even a microwave exposure will cause the depolymerization of these molecules, and which will give the ease for its viscous nature. So even biological modification means we use enzymes. So intervention of these enzymes will change the either the structural components of this biomolecule, that is polysaccharide, or it also have a depolymerizing activity with hydrolases. So that will lead into the modification of this polysaccharide. Next is a chemical modification. So it's a huge and industrially feasible part where the modification will be done in various capacities, where whatever the end products we require, accordingly we modify. And it can be a sulfation, carboxymethylation, acetylation, phosphorylation. So like that, there is a lot of modification we can do for polysaccharide for its usage. So again, sorry, here it's just a structure I have want to give you, and to appreciate like how a simple change like sulfation, phosphorylation changes. And this change of like there will be a hydroxyl group around this polysaccharide most of the times. So that will be targeted for the modification. And if it is a carboxyl methylation, that monochloro acetic acid will be used. If it is acetylation, the acetic anhydride will be used. So we know that how the, and also it can be controllable. So the chemical modification are most controllable, and you can access like how much degree of derivatization you require. So this is a different method, and the structure being given here. So chemical modification, why we need a chemical modification? As I told, most of the bioactive polysaccharide, even if it is bioactive, it cannot be used because it has a very poor water solubility. For increasing the water solubility we need, and the increase of polysaccharide of this solubility will be always done either by introducing the ionic group or hydrophilic groups. As I told you, like carboxymethylation, hydroxypropylation. So these are the like straight away you can do to this polysaccharide, keeping intact its bioactive nature. The modification can be done, and this will easily enter into a solution. For example, one example I want to give, like cellulose. Cellulose is highly insoluble, especially in aqueous medium like water. And when you do complete carboxymethylation, it is highly soluble. Okay, so this type of changes we can make, and we can use a huge application of this polysaccharide. Okay, this is just a pictorial representation where hydrophilic groups, when it is been increased, the water solubility increased. Again, in physical modification, it is the same. Like it cleaves rather than the functional group being added in chemical modification. Here, it will be fragmented, or we cannot call it as degradation, depolymerization. We can do, and such depolymerized fragments will easily can go in the water. So you know, gamma radiation, ultrasonic disruption, even when we want it very lightly, microwave exposure or ultrasonic sonication will do all these physical modification. So next we'll just move on to how this immunomodulatory effect, especially with the plant polysaccharides. See, this plant polysaccharide is the one which has been studied extensively, and which has been shown beyond doubt it can regulate a immune system. So, and it cannot regulate by only virtue of in one way. It can be a multiple ways, and in various routes it can do this immunomodulatory activity. So not only like it induces or activates T cells, B cells, or macrophages, even they also activate complement systems, as well as and produce a lot of cytokines, chemokines, and regulate very effectively the immune system. So it has a multifaceted approaches for this immunomodulatory to act upon. So again, as I told, how it do the immunomodulatory. So most often they have shown it is through generation of ROS or secretion of cytokines. In turn, cell proliferation also occurs. Phagocytic activity of macrophages also been shown. I'll show you more detail in the work which we have also done, the way it depicts. So how this interacts, how these ligands or polysaccharide will interact with the these immune cells like macrophage cells. Especially, I have taken most of the time macrophage cells into as an example here. So we know that generally, I think somebody is writing and all. Please avoid in between.
Sorry, I think it went off.
These TLRs, toll-like receptors, the one which is a key sensor for this immune response to happen. And we generally, if you just, I'll tell you like what are TLRs. These pattern recognition receptors. As I told you earlier, like there are 10 toll-like receptors in humans, which has been classified into 2 subgroups. It is depending upon like where they are situated. Like if it is situated in cell surface, so the most of them are situated in cell surface, that is 1, 2, 4, 5, 6, and 10 TLR. And it recognizes the PAMPs or DAMPs. So this, which is the part of lipids and bacterial proteins, which are the patterns which will recognize by these TLRs. And but when it comes to endosomal or which is intercellularly located TLRs like 3, 7, 8, and 9, which will primarily respond to the nucleic acids, especially from viruses and bacteria, and also from other source. So this is just a general thing about toll-like receptors. So next, like a variety of ligands corresponds to these TLRs have been identified so far. Sorry, like TLR2 is been shown to recognize a peptidoglycan from bacterial cell wall along with the lipoticoic acid. And when it comes to TLR3, which will be in endosome, recognizes double-stranded RNA. If it is a single-stranded RNA, it will be recognized by the TLR7 and 8. So main focus is about TLR4, which is well known as a sensor of lipopolysaccharide, especially from the bacteria. So this is the one which recognize most of the even the plant polysaccharide. And there are other TLRs. I don't go into the depth of that. For example, bacterial flagellin will be the ligand for TLR5. So this is how variety of ligands corresponds to different TLRs been identified, and still it is going on. More work is still undergoing in this aspect. So now we'll restrict to like how this mechanism of immunoregulation of especially plant polysaccharide occurs. See, this plant polysaccharide will activate macrophages by recognizing or binding to specific receptors, especially in the surface, which in turn down the what happens is this in turn makes an immunomodulatory effect. So what are the like main components or main receptors which will recognize especially the plant polysaccharides? Or as I told, TLR4 is the main one, and most of the plant derived polysaccharide will recognize this, and it will attach to this receptor. And next is CD14 and complement receptor. This in a complex, most of the polysaccharide when it binds to this, there will be a change and immunomodulation happens. Next is a scavenger receptor, mannose receptor, and dectin-1. These are the 5 which has been well studied about the plant polysaccharide interaction with the macrophage. See, this is how, which I am showing here, where TLR2, TLR4, mannose receptor, this complement receptor 3, and the scavenger receptor as well as dectin 1 is being situated in the surface. And this can work individually or collectively. So to make a immunomodulation to happen. So we'll see how this each receptor has a significance in one point. Like, for example, TLR4. So this how this interaction, whenever there is a binding, happens. So what happens? The formation of complex, especially with respect to cytoplasmic domain, there will be a complex. This complex happens with the adapter protein mid-88 as well as interleukin receptor associated kinase, and this in turn activates TNF, TRAF, and finally initiates the protein kinases signaling pathway. So this is how the signal cascade happens whenever there is a polysaccharide being a ligand for TLR4. If it is CD14 and CR3 mediates the activation of macrophage. So here in dual combination it happens, like TLR4, CD14, dectin1, TLR2, and CD14 CR3 form complexes for such signal transduction to happen, and this with the pairing and cooperation, it brings out the activation of this macrophage. Again, CD14 is known to be high affinity receptor, especially for LPS activated macrophages. So this is the one which has been mainly studied, and CR3 is a receptor for complement protein and beta glucan. So I'll just take 1 minute to explain about complement system here. Complement system, especially C3. This C3, whenever there is a response, what happens? This C3 will cleave, and C3b will be recognized by CR3. And what happens? This will bring out the complete opsonization of whatever the antigen or bacteria which is there. So this complement also leads to antigen antibody complex, as well as inflammatory response, as well as even it has a cell lysis. So it has a classical pathway, alternate pathway, and also there is a one more like lectin pathway which runs through this complement system. So next, third one is scavenger receptor mediated activation of these macrophages through plant polysaccharides. So this is a very important because these receptor has a multiple ligand binding sort of interaction happens, where that is why here it is been given a scavenger name, where it clears the pathogens, post defense, and signal transduction happens as usual. So this is also one of the receptor being recognized by plant polysaccharide to activate the macrophage. And how it happens? This scavenger receptor along with CR3 activate phospholipase, and this again cleaves and activates the protein kinase. So these signal will be transferred, or signal transduction happens via this mode. So each receptor has a little diversified mode. Also, like all the things, whichever response internally it leads to this kinase activity, and kinase signaling pathway will be activated. Okay, so this is about the third one, and this is about the fourth, like mannose receptor, which is very important because this receptor will resembles lectin protein, and where there is, lectin is a protein which binds a lot of carbohydrate moiety, and carbohydrate binding protein, well known. And this is especially recognized mannose as a residues, and which can also recognize glycosylated molecules which contains a trehalose group or an N-acetyl glucosamine residues. Trehalose, I think we know that trehalose is a part of insect lymph which circulates in the insects, as well as it is also there in the some plants like mushrooms and all, where this is very unusual. Like here it's a disaccharide having one link. It is very unusual type of carbohydrate, and this will be recognized by these mannose receptors. So again, the fifth one is dectin. Dectin also recognizes glucans like beta-1,3 or beta-1,6 linked glucans. See, in glucans, there are many glucans been identified, and it also differs from source to source. So the bondage also differs from source to source. Even if it is a same glucan, the difference between the bondage or a change in the whole structure conformation. For example, it may be having a what we call as a helical structure, beta helical, alpha helical, and all. It all depends on the structure of these glucans, and this is very much important for respective function, or even if we take in immunomodulation, these molecules will have a impact about its structure function in this aspect. So it mediates phagocytosis too, and also along with that immune response of macrophages. So what happens upon activation of these macrophages? TLRs recruit TR domain adapter proteins for the subsequent activation, as I told. So downstream signaling happens, and this I told this mid-88 and various signal transduction which leads to NF-kappa B activation. This NF-kappa B will enter into the nucleus, and this in addition, this TLRS gives rise to activation and maturation of dendritic cells, because I'm talking about the plant polysaccharide, how this is multiple level it reacts with the reacts in activating such immune system. So and pro-inflammatory cytokines also will be produced along with the chemokines, and later on it proliferate and differentiate helper cells like T helper cells, Th1, Th2, which again will lead into adaptive immunity. It regulates the same. So I'll be just moving towards the end section of my talk, like where our own studies, where we have done some immunomodulatory effect of a plant polysaccharide. The first one is like we have seen previously, it was shown that polysaccharide having galactose, arabinose, xylose, these will only leads into immunomodulatory function. But in 2011, we showed like when we tested the high fructan, especially from the garlic extract, we could see the tremendous immunomodulatory activity with respect to this. So this is the first time we have shown, and it's a first time anybody who has really reported about like fructans being given such a immunomodulatory activity, especially from the aged garlic. And this paper has also been cited in, in so many other works. So this has given, this is considered as one of the important work too. So next, this is about a high fructan, but coincidentally, when we were purifying this, especially the fructo-oligosaccharide, oligosaccharide part of the same, we tested with the like together about all the fructo-oligosaccharide. There was a difference between isolated fructo-oligosaccharide immunomodulation as well as the one which we have pooled, or the cocktail sort of fructo-oligosaccharide. So here what I wanted to emphasize on, especially DP3 and DP4, which had given a very good results towards the immunomodulation. And it has been reported, I think in 2015, that the small part of only having 3 residues or 4 residues will also give a immunomodulation activity. Next, very contradictory to the previous 2 have seen, we started testing pectic oligosaccharide and pectic polysaccharide to see which one is more immunomodulatory, which one is potent immunomodulatory. So we have taken this pectic polysaccharide especially from the chickpea husk. So there were not much work, and nobody has done detailed studies with respect to chickpea husk. So we isolated this. Hello, I think
Hello.
in the
Hello, sir. So excuse me for that. Hello, I request all the participants to mute their microphone, please. It's going to give a background noise. Please, I request all the participants in the Zoom link to put off their microphone.
Yeah, thank you, Dr. Pramod. So I'll continue. Like here, we have done 2 things. One is we have done the hydrolysis of the pectic polysaccharide, as well as also we have isolated in 2 forms. One is by virtue of EDTA extraction, and one is by ammonium oxalate extraction. So we have seen that 2 extractions are very different from each other. Because just I wanted to give a like make a note, like any extraction method from the same source, a little change in the condition will give you a different sort of polysaccharide. This has been reported by many others also. So this is a very in plant polysaccharide, when you start isolating the polysaccharide, you will get a different molecular size, different with a different structural components when you change the condition. So this is the beauty of plant polysaccharides, and which leads to different functional attributes. So I'll continue with this. So when we started doing immunomodulatory activity for this both pectic polysaccharide and pectic oligosaccharides, we have checked the proliferation of macrophages with the raw cells. Here we could see it was not very like proliferation was moderately a little high, but it was not exuberantly high. So what we saw is cell proliferation was there, and especially, of course, the pectic polysaccharide. The polysaccharide component was giving more when compared to oligosaccharides. Again, we tested for nitric oxide production, and in two time intervals like 48 hours and 72 hours. Here also, for our surprise, there was a huge production of nitric oxide, especially in the both 48 hours and 72 hours, but it was declining later on. So, but along with that, we have seen phagocytic activity where compared to LPS, this was more phagocytic activity was shown, especially for polysaccharide compared to oligosaccharide. Oligosaccharide was much better than LPS. So here I wanted to give you, like, it was not pre-LPS induced activities which I am showing. It is exclusive, like our molecules which we have want to test, the directly we have done not through LPS induced initially, which many of the workers will do. But when we saw the cytokines production, we have seen activities, but not as much as the LPS. But it was having a good sort of cytokine production we could see, which is significant, and for comparatively to again here polysaccharide was giving a very good results. In the end, we did some Western blot analysis to see how much is the inos like production. Even though nitric oxide production was very much, but when we saw the synthase expression, the synthase expression was relatively low when compared to LPS. So this was again when we compare that ammonium oxalate and EDTA, especially with the both polysaccharide, ammonium oxalate isolated, because as I have told, the structure is the one which is very key. The structure, if differs by the virtue of isolation or extraction, changes the activity. So what we observed is the polysaccharide was more immunomodulatory than the hydrolysate, and the presence of, especially when I have not shown the structural elucidation, but we have done with all NMR and other data, where xylogalacturonone with relatively more galactone. So that will be the possible explanation for the immunomodulatory activity over here, other than galacturonic acid. So this activates in turn in NO production as well as expression of inos, very similar to LPS. So this investigation, again, first report on immunomodulation, especially from the chickpea husk polysaccharides. So this we have just, I'm comparing with the other reports also recently. So this goes, which I am showing here, like it mediates through TLR and TLR2 and 4, activates downstream signaling, in turn activates the transcription factor NF-kappa B, becomes stimulated, and it enter into the nucleus and binds to the DNA, and rest of the synthesis of important mediators will happen, like TNF alpha and others. Again, this will in turn increase NF kappa B to gets activated. So we know that NF kappa B it has a more than around 400 stimuli which will be responded by this NF kappa B, and still there is a lot of. So this is a central transcription factor which will be the one which gives a lot of activation to happen. And very similarly, even for plant polysaccharide, it is the same. And this activation of adaptive immune response further will enhance the overall defense mechanism. So this is the overview of the work which we have done, as well as the generally the polysaccharide, how the immunomodulatory activity will be attained. So I have just one slide for ending, like what will be the future perspective? Like the most of the structure and function polysaccharide is still not completely understood. So it is still, there is a lot of avenue to work with the structure and function relationship, because that is the key for any physiological function of this polysaccharide. Second, inflammation or inflammatory mediators released in macrophages. Because a plant polysaccharide, all the plant polysaccharide doesn't have a very mild sort of inducing effect. It may have lead to many excessive inflammation in the body too. So it should be like thoroughly studied before we really administer or we give as a therapeutic immunomodulatory agent. So again, we know we have to get balance with pro and anti-inflammatory factors, and even plant polysaccharide has worked out as a anti-inflammatory factors, especially in cancer therapy and all. So there is a lot of scope for this plant polysaccharide to bring about the equilibrium about the body, which may not cause harm by excessive activation of this macrophage. So third is, and the last one is, unlike microbial this thing, plant polysaccharide is very safe, but still we have to do the, whenever there is a like a purification, we have to be very careful before going for immunomodulatory activity, whether there is any endotoxin contamination which will be there in the plant polysaccharide, which is been isolated and elucidated for further studies. With this, I thank you all for patience listening. If anything is there, a couple of questions I can, I'll happy to take.
So thank you so much for your elaborative view and the extensive definitions of the involvement of polysaccharides in modulating immune responses. Because earlier it was presumed that only the proteins play a key role in modulating or bringing the immune responses. But the importance of polysaccharide was, or the carbohydrate was very much neglected. And your work has added a lot of interest on the perspectives of the possibility of polysaccharides as the most immune modulating agents, especially the gallic fructans, and then their ability and the signaling to induce immune responses. And this has added a lot of information to our participants. Now I request my colleague Dr. Shankar to coordinate the question answer session. Shankar sir, please.
Thank you, Dr. Pramod. Sir, it was an excellent talk, giving an overview about immunology and linking this presentation to role of polysaccharides in immune regulations. So there are few questions. There are plenty of questions raised by the participants. So there are few questions that I would like to ask you for the participants. The question is, the These carbohydrates acts as antigen, and these are all immunogen, and is they are immunogens.
No, no. This usually acts as a immunogen rather than antigen. So that is well established. So this comes under immunogen. See, for example, previously microbial polysaccharide, they were sometimes considered as antigen because as I was mentioning about, there was an endotoxin contamination along with this, and actually microbial polysaccharide is the one, sometime it also acts as an antigen. Otherwise, plant polysaccharide is completely an immunogen.
Is this carbohydrates interacts with macrophages?
Yes, yes, that's what I told. No, I have shown uh, the receptors, which like TLR4, dectin-1. These are all the one which activates macrophage by binding to these receptors, Manus receptors, scavenger receptors. That's what I mentioned the same. Like these are the receptor which will involve in activation whenever there is a polysaccharide sort of activation through this macrophage.
Okay, sir. So even the participants wants to know that. So okay, can you please kindly share that which of the polysaccharides which are important, especially in immune, that which acts as a immune modulators, apart from the garlic.
That clear cut complete, like, see, this polysaccharide, I think everybody will be consuming directly or indirectly, like through your daily food. So it is just for example, we have seen garlic and onion everybody will be using. So by virtue of that usage, you will be having a like immunomodulatory polysaccharide along with that. So, but there are several reports like immunomodulatory activity having polysaccharide, but still clinical trials and all, it has not completed, and we don't know like the side effects and many more things. So still it is in, like, we cannot clearly say this is a complete immunomodulatory polysaccharide which can be consumed in so much dosage and all, which give you immunoboosting and all. Only thing is anything directly or indirectly in complex, it can be consumed. It obviously gives uh, immunity to the patients as well as for the normal individual.
Okay, thank you, sir. I have a personal question.
Yes,
Since we have all know that right now we are in a COVID-19 pandemic situation,
right.
where the doctor will suggest to go for protein supplements, zinc, probiotics, etc. So what these carbohydrate roles, especially the people who are suffering from this corona infection?
See, I'll tell you, like, um, I mean, divide of this immunomodulation. Taking carbohydrate is always a good, especially the fiber-rich carbohydrates, because see, the gut is your health. Whenever you have a very good health, means you should have a very good gut. So for gut, the prebiotics are very much important uh, to sustain probiotic activity over there. So this type of fibers, other than protein, vitamin, whatever it is, zinc, that is fine. But along with that, normal diet with a stress-free consumption is always better. It's my personal view.
Okay, sir. Thank you very much for sharing your experience with us. Uh, so could you please kindly unshare your screen and be online, sir?
Okay, okay. Thank you. Thank you, everyone over there, and thank you for listening. And thank you, Dr. Pramod, Dr. Shankar, and all of you. Thank you very much.
Harish sir, thank you so much. Thanks for being an advisor and being being our for sharing your experiences and advising us in both in personal and in research levels. And thanks for accepting our invitations to host this be a speaker, a lead speaker on this session. And we really thanks for being with us for extensive time and sharing your experiences, and you have spared your valuable time with us, sir. Thank you so much.
Thank
On
you.
behalf of, yeah, we thank on behalf of Davangere University, Biochemistry Department, as well as Society of Biological Chemists, Davangere branch. Thank you, sir.
Yeah, thank you. Thank you for giving the opportunity to. Thank you.
Okay, dear participants, we are going to host the second session of today. The second session is from 12 to 1, and the speaker of the second session is Dr. Nagaraj S. He is an assistant professor at Department of Biochemistry, Tumkur University, Tumkur, and he is going to speak on experimental animal models in biochemical research. Nagaraj sir. Hello? Nagaraj sir, can you hear me?
Yes, Pramod, I can hear you.
Yeah, thank you. So you can start sharing your, we'll ask you to share your slides. So can you just wait for another 2 minutes to start the session? We'll just give a break for 2 minutes and we'll just start the sessions after 2 minutes. Thank you. Now it's time for us to listen about our speaker of the day, Dr. Nagaraj S. Now I request my colleague, Dr. J. Shankar, Associate Professor, Department of Food Technology, Davangere University, to introduce the speaker, Dr. Nagaraj S, to the participants. Shankar sir, please.
Thank you, Dr. Pramod. Thanks for giving me an opportunity to introduce today's second session speaker, Dr. Nagaraju, who is an assistant professor at the Department of Studies and Research in Biochemistry, Tumkur University, Tumkur. He obtained his degree and master degree from University of Mysuru, and PhD from Department of Biotechnology, University of Mysuru, under the able guidance of Professor K. Kemparaju. Later, he moved to University of Virginia and later Stanford, USA. Excuse me, kindly participant, kindly mute off. Thank you. So after his PhD, later he moved to University of Virginia, USA, and then Stanford University, Stanford, USA, to pursue his postdoctoral fellowship. So currently he is holding assistant professor position at Tumkur University since 2010. His field of research interests are protein chemistry, inflammation biology, immunomodulation, and wound healing. And currently, so he's having 3 research funding projects from Department of Science and Technology, University Grants Commission, Government of India, and Vision Group of Science and Technology, Government of Karnataka. So he has received many awards. So just to mention a few, received Young Scientist Award from VGST, Government of Karnataka, received Visiting Faculty Fellowship to Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, a recipient of Indian National Science Academy INSA Summer Fellowship, and he has published more than 25 research articles in peer-reviewed journals, and is authored 5 book chapters and 1 review articles to its credits. Currently, he is guiding 4 students for PhD degree at the Department of Biochemistry, Tumkur University, Tumkur. With this brief background, so I will invite Dr. Nagaraju to share his knowledge on experimental animal models in biomedical research. Dr. Nagaraj, kindly share your slides. Okay, thank you.
Yes, Shankar.
Okay, over to you, Nagaraj. Thank you.
Thank you, Shankar, for the introduction, and I thank Davangere University Vice Chancellor, Registrar, all the faculty members of Department of Biochemistry of Davangere University, and I thank Dr. Pramod, Dr. Shankar for giving me an opportunity to speak in this webinar series. Today I will be speaking on one of the broad aspects of the biochemical research or biomedical research and use of experimental animal models in this biochemical and biomedical research. In this topic, I will be speaking on different animal models, why we need the animal model, what is their need, and a couple of disease conditions, and what are the animal models that we use, and what are the upcoming animal model in the COVID situations that we can use for to study the effect of the COVID virus. So I'll be concluding with that remarks. So start with purpose or different. We can know we have not left any animals for the experimental purpose, starting from the small mice model to that of horse, cattle, sheep, fishes, right? Monkey, then the birds, the rabbits, ferrets, then pigs, all the animals we have used for the different experimental purposes. Models. The question arises: why we need the animals for the study? The biochemical research describes a broad area of research that comprises studies of. See, there are different fields, that different things we need to do with our biochemical research. To check whether it's going to work to cure the disease condition or not, and it has to bring back the normal, both mental and physical disease conditions to the normal conditions. So studies on the process affecting disease, and totally we want to have the human well-being, the final concept that we want to know. Then the question arises: why do we want to use a different? Why do we want to use animal model for our study purpose? The animal models are used in research when there is a need to find out what happens in the whole. See, if I use a tissue culture or isolated organ systems, so I will be knowing only what happens to that specific tissue or what happens to that specific cells whenever I use different stimulants, but I will not know what is the integrated components, what is the integrated effect of these components. Like in our system, in our body, the organs doesn't function individually. I think the organs doesn't function individually. See, it is a coordinated functional entity as a whole. So the skeletal, the circulatory system works with circulatory system works with respiratory system. Respiratory system works with the skeletal system, muscular system. Muscular systems needs the energy from the digestive system. After the digestive system, it's going to be excreted to respiratory system. So all this has to be coordinated by the nervous system. So it is more of a complex, and some of these systems make the whole animal and functional activities. So we need to study the same replication of these things in some animal models, so we can replicate what are the disease conditions that we are going to observe uh, in the human beings, in the animal models, and in turn we can study the changes happening in the different systems and possible treatment arised from these conditions. So we need an animal model to study any disease conditions or to find a treatment for any disease conditions. So the thing is, animal models are often used for directly predicting the human responses. So what is human response to that? So in most of the cases, it is difficult to find in most of the cases. It's not possible to replace the use of living animals in research with alternative methods. So I will come to that in when we are speaking about one of the disease conditions where it is difficult to find the animal model itself. So first thing we need to animal model to advance in scientific understanding. Biological research helps us to understand how living things work and apply that understanding for benefit of humans as well as the animals. Why animal model? Many basic cell processes, many basic cell processes similar function similarly in all the animal models or the bodies of the animals. Like if I take the breathing, digestion, the movement, skeletal functions, the vision, the hearing, the olfactories, and the reproduction. All these are going to be the same. The functional things are the same in most of the animal models, most of the animals. So you can replicate the similar kind of functional activities in humans, and I can observe the same functional activities in mice. I can observe the same functional activities in rabbits. So I can find the same functional activities in guinea pigs, and the similar kind of functional activities I can observe in different animals. So I can use those animals as the animal model for our research purposes. This is to treat the body works. First, if I understand how the normal functional activities are going to happen, I can induce the disease conditions, and I can look into that how these normal functional activities are going to affect it. So now I can correct that by using some vaccines or some drugs or some chemical molecules which can bring back the normal functions of these systems, so that this can be replicated in the human systems as well. So you are looking at the one side we have got the human system, on the other side the system which is going to be present in the mice model. So if I look at taking away the external appearances, if I view to the internal organ systems, of this animal model, if I consider human as one animal and mice as another animal, if I look at the internal organ systems which are going to be the working functional activities, we can observe so many similar things between these two models. So we can observe the lungs, heart, liver, kidney, intestine, and the bladder. And if you closely observe that, if I don't see the it's a human and it's a mice, both are one and the same. The organs which are going to present look similar. So whatever I want to do whatever the experiences that are going to be exhibited in the human, the similar kind of response we can get from the animal model of using the mice. So first one is animals can act as models for study of human disease conditions. For example, we humans suffer from so many disease conditions. Do animals have these similar kind of the disease conditions. So for example, the rabbit suffers from atherosclerosis, emphysema, and birth defects. The dog suffers from cancer. It suffers from diabetes. Bleeding disorders like hemophilia. Cat suffers from visual impairments. So anything that's going to be problem with the vision, that's a similar kind can be replicated in the animal model of cat. So animals which are altered to create models of this condition called as induced models. So I can find the some disease conditions in the animals, and in some animals I can also induce some disease condition in the animals so that the response from the animals will be similar to that of the human patients. For example, surgery which damages a particular section of a spinal cord in rats gives rise to symptoms like this in human patients. So whatever we observe in spinal cord damage, I can induce the similar kind of disease condition or damage in the um, in the rats as well. So if you are studying like stroke, which is something related to the nervous central nervous systems, we can also do the blockage of the brain in the animal models, and we can induce a similar kind of stroke in the animal models, and we can study what is the response to different drugs which are going to be relieved from the strokes. So saying all these things, two things. So in recent advancement, there is something called as genetically modified animals, like genetic gene technology has allowed development of transgenic animals, where we can introduce some genes into the animal models, and we can induce the disease conditions in the animal model itself, like the human gene implicated in conditions like Alzheimer's disease, or we can implant some tumors. So possibly earlier speakers might have spoken about implanting the tumor into the animal model and studying the different drugs to relieve from the tumor. We can also do the similar kind of studies like obesity or diabetes in the animal models. So what is the outcome? Why we need to do this? Inducing a disease or making a transgenic animal model. Why we want to do this? So the main thing will be to develop and to test the potential form of treatments. Once the researcher learns more about a particular disease, animals are used to develop and test these potential therapies. For example, medicines for any medications that we are going to take, any tablets that we inhale, any vaccines that will be injected to us, or possibly any injections that we take, or uh, any surgeries that are going to be done today will be are successful because this has been uh, first tried, tested, and been successful in the animals. So whatever the diagnostics tools that we are going to use, like scanners, implants, heart pacemakers, artificial hips, why they have been safe and effective and successful? Because this has been tried, tested, and been efficiently replicated in animals, and the similar has been done in the humans as well. So finally, to the last point, fourth one, why we do all these things using the animals, transgenic animals, normal animals, different animal models. We want to protect ourselves, and we want to be safe. Safety of people, animals, and the environment. A new medicine initially tested in vitro, that means in the test tubes, using tissues and isolated organs. Once they are going to be suitable for animal experiments, animal models, they are going to go for the clinical trials. Once they are going to be through the animal models, then they will go for human trials. The animal tested for dose, efficacy, and safety. Once they have been tested, the later on they are going to be tested on the human patients. So once we do the animal study, so that's why we want to do the animal studies. How they are going to be studied. There are two methods that usually been done by the researchers using the animal models. One is observational studies, second one is experimental studies. The observational studies is more of how the animal responds, the animal behaves. This is something we can tell it as hmm, a phenotype, like how it's going to respond, uh, how it's going to show its response by looking at the external features, like whether it's after taking this drug or inducing this drug, whether the movement of the animal will be normal, whether the if I use a mice, whether it can climb to the ladder, or whether it's going to go around the cage in the normal pattern or not. So that is observational studies. Experimental studies is attempt to directly control selected variables, like drawing the blood and doing the parameters of the blood studies, or collecting the urine and feces and looking at the digestive pattern, and and looking at the animals, like scanning the animals, what happens to the different organs. Those things will be studied like experimental studies. So again, if I come to biochemical research, uh, so many different animals will be used here, including from fishes to the birds to mammals to amphibians to reptiles, to all the animal models that we use. So how we are going to select an animal model? How we are going to select an animal model? Whether I want to do a disease condition in mice, whether I want to do a disease condition in rats, whether I want to do an experiment in dog or sheep or cow or a horse. How we are going to select an animal? So the animal should be closely reproduce the disease or conditions under study. This is very much important. So if I want to select an animal that should replicate the disease conditions that we are going to observe in the host. Say for example, if the humans are suffering from a disease conditions, and if I want to use an animal model has to have a similar kind of the characteristic features that we are going to observe in the humans as well. Then only I can use that as animal models. If I don't have the characteristic features that are similar to that of the human, there's no point in using that animal as an animal model for that specific disease conditions. So it should be reproduce the disease or conditions which are going to be under study, and it should be easily available to many researchers. That is not a rare or exclusive animals. The animal model that we are going to use should not be a rare animal or extinct species. I cannot do a research on an animal using a cheetah, no, because the this is extinction. I cannot use a rhinoceros as the animal model, because like single horn rhinoceros, which is at the edge of extinctions, and I cannot use the animals which are going to be very much rare, and there is some exclusiveness as the animal models are going to be used. So that's the second point. And in case of vertebrate model, should be large enough for multiple biological samples. If I want to do blood sampling, if I want to draw the blood, if I want to take out some tissues, and if I want to sample in film for randomly, so I have to use some bigger animal models. So if I want to draw the blood, something like 5 ml or 10 ml in an interval of 15 days or 1 month, so I cannot use mice as the animal models, because mice will not will not permit me to draw the blood of 5 ml or so for my studies. So I have to upgrade my studies to animal models such as a rabbit, so that I can draw a little bit of more blood and tissue samples too. Should fit into available animal facilities of average institutions. This is with pertaining to the research labs, some of the research labs, at most of the universities. So if you're working at the universities, we know the animal facility, even if I want to do an animal on a dog, or if I want to do an animal on a sheep, our animal facility houses may not be able to fit these animals also into the animal house. So I cannot use those as the animal model. So I have to think before designing an experiment for animal model, whether that animal is available at my animal facilities or not, should be easily handled by the investigators. This thing is I'm a little bit ambiguous about this, because once we into the biochemical research or biomedical research, the investigators will be given training to handle the animal. So easily handled means, if you have facilities, something like the animal house, people are going to you handle animals, then it is fine. Should be available in multiple subspecies. We know mice is one of the animal model which has been used extensively for its research, because we can find so much amount of subspecies which are going to be observed, and similar kind of experimental designs can be done with these subspecies. Should survive long enough for results to be meaningful. Yes, this is very important. If I want to design an experiment for a study of 6 months, and the lifespan of the animals or the time period of the animals which is going to give the response is going to be only 3 months, then I cannot use that animals for my studies. So it should be long enough so that it gives concordant and repeated studies. If I do, it gives the similar kind of response. Sufficiently robust. This is also one of the things. See, if I want to inject a drug of, say for example, 1 mg or 2 mg per kg body weight, and if the dose is very high, so that the animal doesn't respond to the dose, so I have to use the robustness of the animal model. So which animals? So if I look at the different animal model that has been used, so there are two majorly we can classify based on their size: the small animal models, mid-sized, large animal models. So small animal models like rat and mouse, mid-sized the cat and the chicken, the large size animal like cow, sheep, pig and dog. Overall, if I look at the laboratories which are going to use these animal models, so more than 50% of the research labs use mice as their animal models, because if I study the genetics of the mice and the response for a disease condition, or response for a drug, or response for an external stimulus of the mice is very much similar. 99% of the response which is given by the mice will be similar to that of the response given by the humans for anything. So for immune systems, for the resistance, or for the whatever the response that are going to be given by the mice will be similar to that of the humans. So mice easily handled, can have subspecies. So whatever the characteristic features I told in the last slide, all are going to be in coordinations with the mice model. So most of the labs use the mice models, and followed by rats and amphibians and fishes, and rabbits, guinea pigs, and some non-human primates. Camels will be used. So these are different disease conditions for which then heart-related circulation, anatomy, drugs, toxins, biology, cell biology, endocrinology, anything, psychology to behavior, infections to genetics, they use animals as their models for the different disease conditions. So way back when it started for the biochemical research, this is one of the paintings, very long back, an experimental on a body pump. Lot of background noise.
Hello, dear participants, could you kindly mute your microphones? Because we are hearing a lot of background sound here. Thank you.
Okay. Thank you, Shankar. Okay. So this is the art in 1768. So you can observe the people. They have taken a bird for some experimental purpose. These multiple reactions. So in 18th century, so you can look at the different response. Some people are not willing to look at that. See, they are not willing to look at that. Some people are very much curious what happens to the bird. So there was in 18th century when they started with animals, they used the models for the study. There was mixed response. Like they were shocked to hear that animals will be used for the studies. There is sadness, appreciation, curiosity. All these were observed initially when they used. So the similarly in 1832, after 1832, when they used uh, physiological demonstrations, vivisection of a dog. Now, even though dog is suffering from a lot of pain, the look at the curiosity of the persons around who are looking at the vivisections, right? So rather than that of the pain or the response for animals, the people were curious to know what will be the response from the animal model. What will the response to animal models? So this is full page illustrations of Pasteur. We know Louis Pasteur's work, and vaccines for rabies. So all this we know. This is full illustrations of the Pasteur in his animal facilities, published in 1884. So at that time, there was a moderate curiosity on Pasteur's work in the US, which would intensify after his first successful human trials of therapeutic vaccines for rabies in 1885. In this, the reader is assured that the use of dogs is both human and justified in the interest of mankind. So we can use so from 18th century to 19th century. So there was like, yes, the animal models can be used. Animals can be used for disease conditions, for the survival purpose of the humans and well-being of the whole systems. So if I look at the more of the milestones, development of anti-rabies vaccines, development of syphilis, insulin, penicillin for treatment of diphtheria, tetanus, hemophilia. So many, all this, whatever we are going to call it as a deadly disease conditions. So all this for the vaccine development, the animal models have given a lot of inputs for developing these vaccines or the treatment for different disease conditions. So these are some of the persons who got a Nobel Prize for their work. So if I summarize all these things, of the 106 Nobel Prize awarded for physiology or medicine, 94 were defined on research using animal models. So this includes every prize awarded for last 30 years. So this says how much the importance is animals will be in the biochemical research. So I will now being known all these things. I will come to two disease conditions. One is diabetes mellitus, and some of the animal models that will be used. Diabetes mellitus is a chronic disease that is characterized by relatively or absolutely lack of insulin, resulting in hyperglycemia. This can lead to variety of complications like neuropathy, nephropathy, retinopathy, the more of risk of cardiovascular disease. Worldwide prevalence of diabetes is 9.2% in women and 9.8% in men, with approximately 347 million people suffering from disease. Diabetes may be classified into two types. One is insulin dependent, and type 2, that is non-insulin insulin independent or non-insulin dependent. Type 1 diabetes is autoimmune destruction of pancreatic beta cells, lack of insulin production. Type 2 is characterized by insulin resistance. Now, do we have an animal model for this study of diabetes mellitus? The answer is yes. And we have more of specific disease conditioned animal models. Say, for example, we have model of diabetes, which are going to be, this is for type 1 diabetes, which can be induced by chemical induction, spontaneous autoimmune modified animals, genetically induced type 1 diabetes, virally induced type 1 diabetes. So we can have these are models: high dose streptozotocin, then Alaxon, multiple low dose streptozotocin, BB rats, LEW, 1AR1, Akita mice. So these are some of the animal models which will be used for studying the type 1 diabetes disease conditions and identifying a possible treatment. for this type 1 diabetes. Say for example, if I in type 1, this one, streptozotocin induced diabetes. So histopathology and immunohistochemistry of the pancreatic isolate of Langerhans in the control panels in the left. This is control in normal, and this is streptozotocin induced diabetes, where the beta cells have been shrinked, or the beta cells have been decreased in very much number. Similarly, type 2 diabetes, we have got different animal models like obese models, induced obesity, non-obese models, genetically induced models of beta cell dysfunction. So we have got different animal models to study type 2 diabetes. So there are many animal models which are available for all the disease conditions. Now the question arises, the pandemic that are going to face right now, do we have an animal model to study this COVID-19 disease conditions? The answer is, right now, we don't have a specific, accurately studied animal model for COVID-19. So the couple of things you might be knowing very much, since we are going to hear this COVID-19 from last December, January, from last 6 months, we have been hearing what is COVID-19 and what is its disease condition, how it's going to get transmitted, whether we have a vaccine for this, whether we have got a treatment for this, when we are going to get the vaccine, and how we are going to get possibly eradicate this disease condition. So there are many questions for this. So severe acute respiratory syndrome, coronavirus 2, SARS-CoV-2, cause infectious diseases. COVID-19, which was first reported in Wuhan, China in December 2019. COVID-19 has now spread to more than 100 countries, and WHO has declared COVID-19 a global pandemic. Source SARS-CoV-2, SARS coronavirus 2 is thought to originate in bats. The problem is, however, the intermediate animal source of the virus is not known. It's unknown. It's originated from the bats, but how it is going to transmit it from the bats to the human system, we don't know this. Now the problem arises. The scientists are still in search of an appropriate animal model. We don't have a exact animal model which can replicate a characteristic features of the humans in the animals. So this is the problem where we are finding difficult to find a treatment or find a vaccine for the COVID-19 attributes to the virus. So is that thing that we don't have, or we are not at the point of identifying animal models? There are several people are working all over the world. The scientists are working on identifying a suitable animal model for this COVID-19. How the animal model should be. An ideal COVID-19 animal model would replicate all the facets of the human diseases. That means all the characteristic features has to be present in the animal models. Right. The characteristic features such as it has to infect the upper respiratory tract, in the inner respiratory tract, has to have the infection in the lungs, has to have the infection in the tonsils, has to have the infections in the it is a kind of pneumonia type of conditions. So all this has to be replicated in the animal models. And do we have that animal models? We'll know that. Hamsters, ferrets, and non-human primates were found to support SARS-CoV-2 replication, display varying degree of non-lethal illness when the virus was delivered into the respiratory tract of these animals. So how to check this, whether this animal model is right, whether I can use this animal model to further develop a treatment for this COVID-19, how I'm going to do this? This I will be able to do. The susceptibility is likely driven by either binding affinity to host ACE2 receptors. Earlier speaker might have spoken about, yes, they have spoken about how the COVID-19 infection happens into the systems, how the entry of this virus takes place. It's a very simple thing. The virus comes, it has spike proteins around its surface. the viral proteins. Now it comes and enter into the respiratory tract, the upper respiratory area. Now the humans have got human angiotensin converting enzyme 2 receptor, ACE2 receptors. Now this receptor is the affinity for this COVID-19 virus. And now this COVID-19 virus binds to that, enters into the systems, and now the replication takes place. That's how the infection is simple. The outer summary of these COVID-19 infections. Now, the animal model should be such that it has to have a ACE2 receptor similar to that of the humans, human angiotensin converting enzyme to ACE2 receptors, and this should have a acceptance for this COVID-19 virus. So COVID-19 virus has to go inside and it has to be accepted by this ACE2 receptors similar to that of the humans. So the one mice, most of the people will know, hamsters, yes. Ferrets, for the student fraternities who are not looking to ferrets, these are ferrets. The ferret is the domesticated form of European polecat. A mammal belongs to the genus Mystella, as the weasel Mystella in the family of Mystelidae. These are ferrets. So the scientists have used different animal models to study COVID-19 infections. So this has been published in a couple of weeks back in Science Journal. Search for best animal models to study COVID-19. They have used cat, chicken, dog, duck, ferret, human ACE2 mouse, hamster, kitten, macaque, and pig. When they used all these animal models, most of the animal models were not susceptible for COVID-19 infections. See here, the cat, chicken, dog, and duck. No response. Pig. The response for the ferrets. Look at these ferrets. Upper respiratory tract infections, yes. Lower respiratory tract infections, yes. Fecal swab virus, yes. Contact transmission, yes. Airborne transmission, yes. Weight loss, no result. So now, all these are going to be yes only for the animal model for ferrets. So looking at taking all these animal models, they come to conclusions that ferret will be the one of the best models to study the COVID-19 viral transmissions. She et al., one of the scientist group in China, they have studied with the ferrets, cats, dogs, and pigs, and chickens and ducks to test the replication, pathogenesis, and transmission of this COVID-19s. So this COVID coronavirus is not new. This has been earlier SARS-CoV coronavirus 1 that has come in 2005, and they have been able to do the replication, pathogen transmission in the model using the ferrets. So ferrets are commonly used as animal model for respiratory viruses that infect humans. So they have used these ferrets and cats for their studies. Sorry. Taken two viral components. One is that the virus that are going to be found in Wuhan region, that is F13E, and the one that is infecting the humans, that the virus that have been collected from the human samples like C10H, okay, isolated from human patients. These two were used for the studies. What's the result? This is the study of replication of SARS-CoV-2 in ferrets. If I look at the slide A, if I look at the picture A, this is for F13E. This is for C10H. C10H is going to be one which are going to be collected from the human. F13E is the one which are going to be found in the Wuhan regions, which has infected the human. See this one? Look at this. Nasal, turbinate, soft pellets, tonsils, upper respiratory tract and lower respiratory tract which are going to get infected, very much high in ferrets. Okay. So A and B will be the F13 and C10. C and D will be the viral titer in different organs. See? Viral titer very much high in ferrets. E and F, viral RNA. E and F. They wanted to check now this virus is going to be inoculated into the through nasal into the animal models. Now, whether it's going to have the replications inside the animal, they wanted to check this. So it definitely replicates in the ferrets, and G and H will be the viral titer. This is viral RNA, viral titer, and I, J, and K and L will be the amount of antibodies that are going to be produced in these animal models against this COVID-19 virus, and K and L will be whether these antibodies will neutralize the virus, something like whatever we are going to speak about, like the plasma therapy, plasma therapy that are going to be possibly using. In India also they are trying to do this plasma therapy, where we can have this raised antibodies in one person will be transmitted to another person, so that we are going to have innate immune transfer of uh, already developed immune system from one person to another person's. This will be the one that tried this. So in ferrets, it's going to work beautifully. They're going to work beautifully in the ferrets. So they thought that ferret will be the best model to study the COVID-19 infections or developing of the vaccine for COVID-19. And if you want to try this vaccine, the COVID ferret will be the most acceptable animal model for the COVID-19 treatment purpose. They wanted to check whether the same can be replicated in the cat as well, because the cat and ferrets have got similar kind of the response for the respiratory infections from the viruses. They wanted to check the cats, because cats in Wuhan regions, they observe similar kind of infection in the humans, as well as the similar kind of viral infections were observed in the cats as well. The cats also suffer from the same conditions as that of the humans from the COVID-19 infections. So they wanted to check whether the cats can be used as animal models. Of course, the cat can be used. The viral, they have taken the cats of juvenile, the small cats, as well as subadult cats for the studies. And they are also shown infections from the COVID-19 in nasal tubercle, soft pellets, or tonsils, or trachea, as well as in the lungs. They were able to show the infections from the COVID-19. When they were checked whether it can be transmitted from one cat to another cat. See, the COVID-19, the animal models can be very much good if you observe the replication of the virus in the animal models, respiratory problems, that is upper infections to upper respiratory tract, infections to lower respiratory tract, infections to the lungs, and infections to other organs, and whether once the one animal is going to be infected, whether it is able to transmit the infection from its one animal to another animal. That is the primary thing that when you are going to use animal model, because now we know that the COVID-19 is going to transmit from one animal to another, one human person to another human person. Whether the similar kind can be observed in animal models. Once the animal model which shows this kind of transmission from one animal to another model, one animal to another animal, that will be the best animal model to study the COVID-19. So when they studied these cats, inoculated and exposed, when they studied that, the exposure was not clearly transmitting the COVID-19 from one animal to the another animal. So they thought when it compares to cats, the ferrets will be the one that are going to be very good when transmitting the COVID-19 from one animal to another animals. So from these studies, they're able to find that ferrets and cats differ. AC2 is mainly expressed in type 2 pneumocytes and serous epithelial cells of tracheobronchial submucosal glands in ferrets. And ferrets and cats differ by only 2 amino acids in SARS-CoV-2 spike contacting regions of the AC2. Therefore, the underlying mechanism that prevents the application of SARS-CoV-2 in the lower respiratory tract of ferrets remains to be investigated. The fact that SARS coronavirus 2 or COVID-19 replicates efficiently in the upper respiratory tract of ferrets makes them a candidate animal model for evaluating efficacy of antiviral drugs or vaccines against COVID-19. Now, from these studies, they were able to say that the ferrets can be used as a best animal model to find any antiviral drug or vaccines against COVID-19 can be tested on these animal models. So, looking into this, the ferrets and the cats. So the cats can be, so you might have heard the news that the tiger in the zoo might be contracted with the COVID-19, whether it is true or not. Yes, the tigers can be, because these are the cats and tigers have got the genetic similarities between these two, and the receptors of ACE2 will be similar in cats and the tigers. So there is possibility that the tiger may also get contracted the COVID-19 virus from the human samples to the tiger. So it is possibility that the tiger will also get the COVID-19 infections. Now, our questions, main concern is whether the pets that we are going to be having in the houses like dog, pig, chicken and duck, whether these have the possibility or susceptible for COVID-19 infections, whether this COVID-19 can be transmitted by dog or can be transmitted by pig, can be transmitted by chicken or can be transmitted by duck. This slide gives some of the informations about that they have done, whether it's going to have the possibility of transmissions. Pig, chicken and duck, if you look at the table, 0 by whatever the number, 0 means they don't have any possibility of transmission, or even if you inoculate the COVID-19 virus into these animal models, they will not show any type of the features of the COVID-19 infection. They are not able to replicate, or they were not able to have this COVID-19 multiplied in their respiratory tract and show the features of this possibility of COVID-19 infection in the animals like pig, chicken and duck. They don't have the we cannot detect any type of infections in the lungs or trachea or nasal trabecular, soft palate, brain, heart, tonsils, kidney, spleen, liver, pancreas and small intestine. Any of these organs or any of these regions doesn't have the infections except dog. Okay, dog, there is no infections in the oropharyngeal swabs, no, but in the rectal swab, in the feces, there is possibility that some of these viruses can be, if they enter into the digestive system, they can also get transmitted from, they can also observe this in the rectal swabs, in the feces can be observed. But whether that can be infecting a new animal, that has been not studied yet. Possibly they might have come with a couple of publications regarding that. So outcome, vaccines and treatment against COVID-19 could drastically reduce the COVID-19 transmissions, saving lives and brightening prospects for economic recovery. But as the pursuit of SARS coronavirus 2 vaccines and antibodies surge on, animal models play the important role to determine the effectiveness of potential therapeutic strategies. The available studies suggest that ferrets, hamsters, and cats may serve as attractive alternatives to non-human primates and transgenic mice. So if you have a mice which are going to have this human angiotensin converting enzyme 2 receptors in the mice, and we can also study this using the animal models. But ferrets will be the best model to study the COVID-19 infections. So looking at the two disease conditions, like one is diabetes mellitus and COVID-19, what is the other possibility of using the third of the animals? We can also develop antibodies. Looking at the disease conditions, developing the disease conditions, treating these disease conditions, what is the other things that we done is raising the antibody, using that antibodies for research purposes, right? Then we generate in variety of species, the antibodies like sheep, goat, rabbit, donkey, or chicken, and different experimental design or experimental purposes. the antibodies that are going to be used for treatment in snake bite treatment, that also can be directly from the animals. If I want to check the disease conditions by ELISA, I need to have the antibodies to do that. Then if I want to do immunoblotting, immunostaining, all this requires the antibodies raised or developed in one of the animal models. So this I will not go into detail, like production of polyclonal antibodies, production of monoclonal antibodies. And if I have monoclonal antibodies, I can do several diagnostic applications like radioimmunoassay, then ELISA, and development or measurement of concentration of hormones like insulin, pregnancy test kits. They do have the antibodies for human chorionic gonadotropins, progesterone, thyroxine. And if I want to check the blood group, like blood typing, concentration of cytokines or interleukins, histocompatibility, all this can be checked using monoclonal antibodies by diagnostic applications. We can also do the imaging like if there is a cancer or if there is a tissue, if I want to image, like I can use some monoclonal antibodies attached with radioisotopes like iodine-131 or technetium-99, and I can use that monoclonal antibodies to identify or to specifically say where the conditions, like identification, especially this will be used to identification of cancer tissues, where the cancer tissues and treatment of cancer tissues, myocardial infarctions, deep vein thrombosis, therapeutic applications. There are many applications of these monoclonal antibodies. Now the question comes, sir, we have some animal at my farm, some chickens or some mice and rabbits. I have some idea. Can I do that at my farm or at my home? Or can I do this experiment by my own? The answer is no. You cannot do that. And we are not permitted to do any experiment on the animal until and unless we are taken permission from CPC-SEA, that is Committee for the Purpose of Control and Supervision of Experiments on Animals. It's a statutory body formed by the Act of Indian Parliament under the provision of Cruelty to Animals Act 1960. There's something called the Institutional Animal Ethical Committee in the research institutions as well as in universities, constituted in every laboratories, which are only empowered to approve the research product. Before doing any experiments, before doing any research projects, if you want to inject anything to animals, if you want to study an animal, the CPCSE and IIEC has to be permitting you to do that.
Hello? Hello? Nagaraj sir, can you hear me? Hello? I can see your slide shared, but no audibility. Hello? Nagaraj sir?
What experiments you want to use? Now, do we have the replacement for that? Yes, we can replace the alternative techniques to avoid altogether. Or the thing is, we can reduce the animals that are going to be used to minimally use the animals, and we have to refine ourselves before doing that animal models, we have to do some several in vitro studies so that we can do the final things in the animal models. Need of the hour. Greatest challenges faced by the modern biochemical research concerns complex, multifactorial diseases such as cancer, cardiovascular disease, infectious disease. There are several diseases that are going to emerge, and we need several animal models for this. And we need to have transgenic animals to do a lot of animal models. And the research on relevant carefully, we have to carefully design well characterized, controlled animal models will remain for a long time for fundamental discoveries. So this is one of the advertisements published in 1991 in the US, one of the journal, the Hour, a part of the campaigning to defend animal research. Humans and animals would still be dying from rabies if Pasteur hadn't experimented with dogs. So this gives that animal models as a must for biomedical research or biochemical research. And with that, I conclude my talk on use of experimental animal models for biomedical research or biochemical research. I want to acknowledge my mentor, my guide, Professor K. Kempraju, Professor Department of Biochemistry, University of Mysuru, who inculcated scientific temperament in me during my PhD studies. I want to acknowledge Pramod for giving me an opportunity in this webinar series. I want to acknowledge my institution, Tumkur University, Tumkur. I want to acknowledge Tumkur University, Vice Chancellor, Registrar, all the faculty members of biochemistry, everyone who have directly or indirectly organized this webinar series. So thank you one and all.
Thank you, Dr. Nagaraj, sir. So you are connected very well with the need of the day, the animal models possible to discover and to invent the possible drugs to the COVID-19. That is a major pandemic that is the world is going through. And then you're given an extensive view on the possible animal models that will be used in biochemical research for understanding the mechanism and to delineate, to, to, to identify the target for various drugs that has been either synthetically or naturally designed. And your talk was very much followed by the YouTube and Facebook, and as well as in the Zoom by many participants. Now the session is open for discussion. I invite Dr. Deshankar, faculty member and my colleague at Davangere University, to moderate the session. Shankar sir, please.
Thank you, Dr. Pramod. Dr. Nagaraj, for an excellent talk on usage of animals for biomedical research.
Thank you.
There were several questions which are raised by the participants.
Okay.
So we have picked a few questions. Could you please clarify those things? Okay.
Sure.
As we all know that whenever we are using animals, it's compulsorily required ethical clearance.
Yes.
So do we require ethical clearance to use fish for the studies?
See, yes, this was one of the questions. See, if you are going to use the fish, now the animal ethical has not bring the fishes into the CPCSIR IASC guidelines. The small fishes. So I have seen so many laboratories which are going to, because these don't need the permission from the CPCSIR IASC for experiment doing on the small fishes. I'm not well versed with the using a larger fishes like the catfish or the shark fishes or something like that. If you are going to use small fishes like whatever that are going to keep in the home, so definitely you can use that as the animal model for your studies, but you don't require a CPCSI or IAC permission to do that. You can do that and you can publish that. Yes, you can use the fishes directly.
Okay, thank you. So the next question is, the results which are obtained from the animal model can be comparable to for the treatment for the human symptoms? Human symptoms, systems, human systems.
Yes, of course. That's why the primary question that we need to ask before doing an experiment is whether the characteristic features exhibited by the human, whenever there is a disease condition, the similar kind of features that are going to be exhibited animal models itself. So that's why we have to select the animal model which exactly replicate the disease conditions that are going to observed in the humans. That's why for COVID-19 infections, it is difficult to find an animal model because we are finding the animal model which is exactly replicating of the animal systems. Now we have got a couple of animal models like ferrets can be used, or non-human primates they are trying to use, or some of the transgenic mice. They are not started transgenic mice. So those can be used. The animal models should be such that it has to replicate the disease condition of the humans. Then we can use it, definitely.
Okay, thank you. The next question will be, can COVID-19 infection affects the nature of B and T cells of a human system?
Sorry, I didn't get that.
Okay, I'll repeat the question.
Okay, Shankar, yes.
The COVID-19 infection, okay, will that affect the nature of B and T cells?
Oh, the B and T cells of the immune systems.
Yes.
The problem with the COVID-19 infections is now we are not yet looked into the immune response from our systems. See, some people, the more viral infections are going to be deadlier for the persons who are above 60 years and below the age of 10 years or 15 years, because the adaptive immune systems in these two persons, like after 60 years of age and below 15 years, the adaptive immune systems is not going to be as high as that we observe in the adult conditions. So if you observe the mortality rate is going to be minimal in the persons who are going to be adult within the age of 20 to 30 or 35 or 40. So if you are looking into these aspects, I don't think the coronavirus, the COVID-19 is doing something to do with the T cells and the B cells, because we are trying the plasma therapy. When we are trying the plasma therapy, we can definitely say that there is no effect on the T cells and the B cells as such now, right? So the good immune system, if you have, you will not be knowing whether the COVID-19 virus has come and gone, and you'll be fine with that. So I don't think the T cells and B cells we have right now any effect due to that of the COVID-19s.
Yeah, precisely. It won't affect the nature of
nature of T and B cells. No.
Yes, yeah. So the next question will be that disorders in animals and human beings are different, but why we are using animal models for clinical trials?
So initially before trying on the humans also will be used as the clinical trial. We also use the humans as the clinical trials. Possibly not in India, but in other countries like US and Europe, we do have clinical human trials will be done, where the group of people will be consent and they will be taken for the clinical trials, of course. But before going to human trials, they will do a lot of clinical trials in the animal models, which we see similar kind of effect and similar kind of response from that. Once it has been cleared through all these animal clinical trials, then they will definitely do the human trials. So you might be hearing in the Italy, they have tried with the COVID-19 vaccines. They are starting with human trials or human studies. So that's nothing but the humans will also be used as the animal models in the clinical trials. Definitely. Possibly it has not come in our countries, to my knowledge. In other countries, they do use the humans as the clinical trials. They do have that.
Okay. The next one will be, can I use some veterinary drug or medicine to cure some of the diseases in human beings, whether by altering concentration in composition or by using some other combination?
This is like this is something like, can I use the veterinary drug for treating the human disease conditions? So something to my knowledge.
Yes, and also with the concentrations.
With the alpha concentration. See, the one thing we have to keep in mind when we look at the veterinary conditions, like say for example, if I use a drug in higher mammals like cow or sheep or something like that, we should know that these animals, we have got lot of immune resistance. When we rather than immune resistance, the capacity of the animal responding to external stimulus will be very much sensitive in humans when compared to these animals, right? Say for example, because the habitat, the food patterns, uh, the system that are going to be in these animals will be different from the human systems. So we cannot use those drugs. So definitely some of the drugs will be used. There are several drugs which are going to be used in veterinary as well as in the human system. But the concentration, definitely they are going to differ. But it cannot be true for all the drugs. It can be true for only few. It cannot be true for all the drugs. And second thing we have to know is the receptors which are going to be present in the humans will be similar in the animals, but not the same. They are similar. They are similar. When they are similar, the response to a stimulus will be different. So I cannot use the drug same which are going to be used in the veterinary to that of the humans. No, I cannot use it. We cannot use it.
Yeah, thank you. The next question will be, how the results can be compared if we use cell lines in place of live animals, live models?
Okay, this I had we discussed in the two second or third slide we discussed. Of course, I will answer it again. See, in cell lines, we are going to take cell lines which are going to be specific from one organ. Say, for example, I am going to use endothelial cell lines, or we are going to use cell lines from specific organs like kidney cell lines, or cell lines from pancreas, like isolate of Langerhans, or muscle cells, or some of the cell lines like nervous systems. So it is not integrated. It is, say, if I want to tell, if I take a tablet, the tablet go to the digestive system, absorbed by the digestive system. Through the circulatory system, it's going to go to every organ, and now the response comes. So it is the drug versus the response is a integrated system. Our human system works by integrated system, nervous system, circulatory system, all these are interconnected. So anything that we give will be responded by all these systems. In cell line, I'm going to study only to particular to that cells, but I will not be knowing whether this effect will be same to the cells of the other systems. Like the circulatory system cells may be fine, but it may not be used the same response from the muscular system, from the respiratory system, from the excretory systems. So I cannot, once all the cell line studies have been done, the drug has to go through the animal studies.
Okay, thank you. So this one will be very interesting. It may be useful. Your answer may be useful to the young research and also the students of PG students. So how one can publish a research work done by animal before it become compulsory to take permission from agencies for sacrification, animal sacrification?
That means the person.
So how can, so because anybody wants to publish a data, so research output.
Yes.
So they're asking whether it is necessary to take a permission, and how can we approach to take the permission?
No, first there are two questions: whether we want to take the permission. Second thing is how to take the permission.
Permission, and also for sacrifice, to sacrifice the animals.
To sacrifice the animals, yes. So, first question is whether we want to take a permission or not.
Yes.
It is big yes. Yes. We need to take the permission to do anything we want to do on an animal model. You need to take a permission. Without taking permission, you cannot handle the animals. You cannot test the animals for any experimental purpose. We need to take the permission from CPC-SCA and IAEC, Institutional Animal Ethical Committee. We must take the permissions. And second thing is where to take these permissions. So every institution, every university, every research lab, every research institute, every university will have a CPCSCA and IAEC committee. CPCSCR, Institution of Animal Ethical Committee. This committee will be formed with faculty from life science, faculty from possibly one from psychology from one from doctors. So they have got one from pharmacy. So they have got 4 to 5 people will be included in that committee, and you need to take the permission. You have to write a project. You have to write a project, something like, this is my work and these are the methodologies I want to use, and this is the type of experiment, whether you are going to feed the animals through orally, whether you're going to inject the drug to its intramuscular route or intravenous route, or whether you are going to inoculate something, or you want to you are going to develop a cancer tissue inside the animal. You need to give the clear-cut methodology to that committee, and committee will look into that project and look into the methodology that you are going to follow. If it is right, they will give you the permission. You can do that. Last one is to sacrifice the animals.
Sacrification and disposable.
Sacrification, disposable. See, sacrification, again, you have to take the permission. The IAEC will be deciding whether the animal has to be sacrificed for your experimental need. If the experiment needs the sacrification of an animal, there is several procedures for sacrificing the animals. So that procedure has to be followed, and you can sacrifice that. And for disposing, they have got procedure for disposing the animals. So you cannot throw the animals after sacrification as we wish. There's a procedure for disposing the animals where the biosafety disposal has to be by bags, has to be brought, and that has to be disposed in. If you have a hospitals or research institutes where they have got disposal area, they have to dispose to that. Otherwise, they have got the procedure for disposal after sacrifice.
Okay, Dr. Nagaraj, thank you very much for all the clarifications which are asked by the participants. There are so many questions, but due to time constraints, so we would like to conclude the session. Once again, I would like to thank you for your participation in our National Webinar Series 2020, organized by Department of Biochemistry in association with Society of Biological Chemistry, Davangere branch, Davangere University. So,
thank you, sir, and thank you, Pramod, and thank you, Davangere University, all the Vice Chancellor, Registrar, and all the members of the Department of Biochemistry, and all the participants to give them an opportunity to be a part of this webinar. Thank you, one and all.
Thank you. So I would like to express my heartfelt thanks to giving me an opportunity to moderate today's session. So I'd like to thank Department Chairman, Dr. Vatapudi Kumar, my fellow colleague, Dr. Pramod S, and the other faculty members of Department of Biochemistry for organizing this wonderful national webinar series on immunity and infection, biochemical approaches to therapies. Thank you. Thank you once again. Thanks a lot. Very much. Thank you.
Thank you.
Nagaraj sir, thank you so much. And with this, we are going to conclude this session. And on behalf of Department of Biochemistry and Society of Biological Chemists, I once again thank you, a big thank you for making your time to give this wonderful presentation. So thank you. We are going to end this session.
Thank you. Thank you.
Dear participants, tomorrow we are going to have one session, the valedictory session from morning 11 to 12. Will be the session 12, and from 12 to 1 we'll have a valedictory program for about 30 minutes. So with this, we'll again meet tomorrow morning at 11 o'clock. So thank you so much. Thanks for your participants and involvement in the webinar series. So we look forward tomorrow. Please wait for the feedback form. The feedback form will be organized and will be sent to the respective groups to your WhatsApp number by afternoon of tomorrow. So you have to submit. There is a time limit for submission of feedback form, and the feedback form has to be submitted by evening 5 o'clock on the same day tomorrow. So with that information, so we will meet you tomorrow morning at 11 o'clock sharp. Thank you so much. Have a great day.